So hello students, welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. So quickly let me know in the chat right now students if my voice is audible to all of you and the presentation behind me is visible to each and every single one of you. Quickly let me know in the chat. Yes. Good afternoon everyone. Good afternoon Gayatri, Pilo, Pavitra, Chandana. Yes, good afternoon each and every one of you. Now students, this is the first class. That's right. This is my first class from the RNA batch. Now, what is the main goal of RNA batch? Let me tell you. The main goal of your RNA batch is going to be complete coverage of the chapter. All the way from the basics, even if you do not know a single word from this chapter, I will make sure that this chapter becomes very easy. The chapter which I am referring to is none other than your photosynthesis in higher plants. Now, I have spoken to many students and they always tell me, so, photosynthesis in higher plants is one of my backlog chapters. So today, students, today is the end of that. Because I'm going to make sure you tell it in the end of the class, after today's class, sir, this is my favorite chapter and this is the easiest chapter of all time. That is my guarantee only if you watch till the very end. That's right. Only if you watch till the very end, then only you'll be able to say, right, this is the easiest chapter of all time and this is my favorite chapter of all time. Now why? Because this is a chapter which will get you easy marks. Easy marks. If you know in your NEET 2024, this chapter, photosynthesis in higher plants, was for 16 marks. 16 marks. One chapter, 16 marks. So can you afford to lose such high marks from a single chapter? No. So students, without wasting any time, let's start today's class. That is start of RNA batch. If you do not know a single word in this chapter, doesn't matter. Because I will be teaching you from all the way from the basics, so you can understand and we will increase the difficulty as we go. And how do you know the difficulty is increased? When we solve the questions. Okay. So everyone in the chat, quickly fire emojis in the chat because this is going to be the most easiest chapter of all time for all of you. Now I want everyone to quickly smash that like button first. Yes. Everyone, quickly smash that like button right now, all of you. Smash that like button. Mark your attendance, all of you. And let's start the chapter. Let's start with the chapter. Now, quickly, first of all, the basic definition. The basic definition of your photosynthesis. Photo means what? Means light. Photo means light and synthesis means produce. So let's understand the chapter with the help of photosynthesis first. Right, photosynthesis first. So if I ask you a definition right now, what is the meaning of photosynthesis? Many of you will tell me, sir, photosynthesis is a method or a process by which plants prepare their own food. Right? All of you would say that, right? Everyone would say, sir, photosynthesis is a process by which plants prepare their own food. Technically, the definition is correct. But that definition is actually a class 8th definition or a class 10th definition. Right now, you're in class 11th or class 12th or class 13. It's a dropper. You cannot use a simple definition. Let's tweak the definition into a much more higher level. Yes. Before we do that, increase the difficulty level of the definition, let's understand what is photosynthesis first. Okay. For us to understand photosynthesis, should we not look at what is inside a plant cell? What exactly is happening inside a plant cell now? Shall we? Yes. So, students, this is your simple cell. Let's take a simple plant cell. Okay, sir. This is a simple plant cell. You know what? Let's make it green. Very aesthetically pleasing it will be, right? Let's make it green color. Okay. This is your simple plant cell. Okay, sir. We have a simple plant cell here. Now, students, tell me in the chat, do the plants take in carbon dioxide or do they take in oxygen? Quickly in the chat. Plants usually take in what? Plants usually take in the CO2. Every class 10 student should be able to tell me, plants take CO2, plants release oxygen. We do the opposite. Simple, right? Now, inside a plant cell, this is your plant cell. This is your plant cell, students. Inside a plant cell, this CO2 is getting converted to inside a plant cell, this CO2 right here is getting converted to something called as C6H12O6. This C6H12O6 is nothing but your glucose. Yes, it is nothing but your glucose, right? Now let's play a simple game. Simple game. Tell me, spot the difference. The game called as spot the difference. Here we have carbon, here we have carbon. Here we have oxygen, here also we have oxygen. But here in the glucose we have hydrogen. Do you have the same hydrogen here? Obviously not. There is no hydrogen there. So can I tell basically from CO2 to glucose there is something called as addition of hydrogen. Yes sir, there is addition of hydrogen here. Addition of hydrogen. So in the terms of scientific, how do you call it? Students, listen to me very carefully. The process of addition of hydrogen is called as reduction process. It's called as what? Reduction process. All the way from the basics will go, students. All the way from the basics. It's called as reduction. So students, that is the first definition of your photosynthesis. Photosynthesis is a reduction process. Photosynthesis is a reduction process, right? It is a complete reduction process. Complete reduction process. Simple definition. Yes, simple definition. Now, I told you this CO2 got converted to glucose. Right, sir? I believe you. Now, but sir, where did this hydrogen even come from? Where did it magically appear from? Did someone add it there? Yes. If you have a garden in your house, do you regularly water the plant? Yes, sir. Plants require water. Yes, plants require water. One of the main reasons is that it is the same water. It is the same water which is providing the hydrogen. It is the same water which is providing the hydrogen for the glucose. Now, if hydrogen is added to the glucose, what happens to the oxygen, students? What will happen to the poor oxygen? Obviously, the oxygen will be removed as a byproduct. Byproduct. Don't we study that? Don't we just study that? Usually, we always learn plants take in CO2, plants remove out oxygen. Now, this is the point. This is the point where you learned something extra. You learned the mechanism behind it. Awesome. Now, let me write a few points here which will help you to understand. The entire process of photosynthesis in a much more better manner. For example, I told you it is a reduction process. But what exactly is getting reduced here? I told you addition of hydrogen is called as reduction. So technically speaking, CO2 is getting reduced to glucose, right? CO2 is getting reduced to glucose. Can I write a point here? Yes. CO2 is reduced. CO2 is reduced into glucose. Perfect. Now students, let me ask you one more question. Do plants require sunlight? Yes or no in the chat quickly. Do plants require sunlight? Yes, obviously the plants will require sunlight. Sunlight is inorganic or organic? It is obviously inorganic in nature. CO2 inorganic, hydrogen inorganic in nature. So plants are technically utilizing all the inorganic substances and getting converted to organic substances. So plants are using what? Plants are using inorganic substances and converting it to organic substance. Inorganic to organic substances. And can I call it anabolism? Yes. So technically your entire plant right here, it is also anabolism. Awesome. Now, is the plant absorbing the sunlight? Yes sir, the plant is absorbing the sunlight. So can I call it a endothermic reaction? Yes, it is an endothermic reaction. Perfect. So what do we learn? From the one slide right here, students, from the one slide right here, every single student should understand you have so many questions from one slide right here, one slide. That is, plants take in CO2, the CO2 gets reduced to glucose, CO2 is getting reduced to glucose. That is, reduction is called as what? Addition of hydrogen. CO2 is reduced to glucose. Inorganic substance is getting converted to organic substances. Oxygen is released as a byproduct right here. And also, your CO2 right here, this CO2 right here, students, extra point, is it losing the oxygen? Is the CO, is not CO2, is the H2O right here, is the H2O losing the oxygen? Yes, losing the oxygen. Is it loss of, can I say it's also losing hydrogen? See, H2O, is it losing, see, hydrogen is going here. So it means for this water, there is loss of hydrogen right here. Yes, there is loss of hydrogen. So loss of hydrogen in technical terms is called as oxidation. It's called as what? Oxidation. So your water is undergoing oxidation, CO2 is undergoing reduction. Any doubt here? Ask me quickly. I believe there should be no doubt in this first page right here of your chapter. And if you understood this right here, this slide right here, you technically understood half of the chapter. Technically understood the half of the chapter is done right here. If you technically understand this. See, CO2 is getting converted to glucose, reduction. Water is undergoing oxidation. Byproduct is oxygen. Inorganic to organic. Done. Done. Now let's understand a few other concepts. The first concept which you need to learn right now is the definition of your photosynthesis. I mean, initially you used to think photosynthesis is just plants preparing their own food, but it is not so. The actual definition of your photosynthesis is that it is a physico-chemical process. It is, if you add one more point here, enzymatic mediated, you can add that. Enzymatic mediated physico-chemical process. What is the physical process here? The physical process here is exchange of gases with the help of stomata. The chemical process we just learned, the chemistry. That is the chemical process. So it is a physico-chemical process in which carbon dioxide plus water in the presence of sunlight and chlorophyll, glucose and oxygen are produced. Glucose and oxygen are produced. Clear? Oxygen is the product. Glucose is the product. Oxygen is the byproduct. Oxygen is the byproduct. Clear, all of you? Clear? Now, plants are capable of synthesizing organic compounds by utilizing the light. Now, one more question for all of you. What do you call this mode of nutrition when plants prepare their own food? What do you call it? All of you remember, right? It's called as autotrophic mode of nutrition. It's called as autotrophic mode of nutrition. Beautiful. Now, plants convert light energy into chemical energy by the process of photosynthesis. We learned that. Now, what is the importance of photosynthesis? Obviously, we know in any food chain, take any food chain, grazing food chain or aquatic food chain, right? Plants are usually there somewhere, right? Mostly when you talk about terrestrial ecosystem or terrestrial food chain, right, grazing food chain, the primary producers are always going to be plants. The primary producer are always going to be plants, right? And responsible for the release of oxygen also, because oxygen is the byproduct there. Clear, all of you? Easy, right? Do you think the chapter is easy? Do you think the chapter is easy? Yes, it's going to be very easy, students. Just follow my words. Follow my words. Write down few definitions. Write down the cycles and write down the conversions. The chapter is going to be butter. It's smooth as butter. Very easy chapter. Now, let's study some different experiments by different scientists which will help us to understand the photosynthesis in a much more better manner. For example, the first scientist right here. The first experiment right here is called as variegated leaf experiment. Variegated leaf experiment. All of you know this. All of you know this. We have a variegated leaf here. The meaning of variegated leaf is very simple. A leaf which has variable amount of chlorophyll. Variegated leaf has variable amount of chlorophyll. For example, wherever we have the dark regions, in those locations we have more chlorophyll. Yes, more chlorophyll. In the light green region, we have the less chlorophyll. Simple. That is a variegated leaf. Now to this variegated leaf, you add iodine. Now one thing missing here is, first you need to expose the variegated leaf to sunlight. Obviously, there should be photosynthesis. Now once you are exposed to sunlight and then you add iodine, you will notice a very interesting result. That is, all the dark green region will turn into blue-black. Can you see? All the dark green region will turn into blue-black or violet-black in color. All the dark regions. But what about the light regions? Light regions will not turn anything. They're as it is. They're chilling, light regions. Now what does it tell you? It tells you that only in the darker regions, only on the following darker regions, photosynthesis has occurred. Photosynthesis has occurred. Photosynthesis occurred. Now why only in the darker regions photosynthesis occurred? Why? Because in the darker regions there was chlorophyll. There was presence of chlorophyll. Simple, right? Presence of chlorophyll. So chlorophyll is necessary for photosynthesis. Done. First experiment done, all of you. Did all of you understand? That first experiment tells you that importance of chlorophyll. That's it. Variegated leaf experiment. If you understood the following experiment, quickly green hearts in the chat, which will help me to identify that yes, you have understood the first experiment. Now let's go to the second experiment now. Let's go to the next experiment. It's called as Moll's half leaf experiment. Again, very simple, very simple. This experiment tells you that you take a plant. On this plant, take one leaf. In one leaf, half of the leaf, you cover it with, half of the leaf, you need to add it in KOH. KOH. Okay, you add in KOH. Half of the leaf is exposed to the air. Exposed to air. So I'll repeat again. Half of the leaf is in KOH. Half of the leaf is in air. Simple. Right? Simple. Understood, all of you? Now what exactly happens here? Whenever KOH is exposed to the particular air, it will remove all the CO2. Every single ounce of CO2, every single molecule of CO2 is completely being consumed by the KOH. KOH will completely consume the CO2. So technically speaking, in this region, there is no CO2, or CO2 is absent. CO2 is completely absent because it is absorbed by KOH, gone. Now what about in the air? Yes, CO2 is present here. In the other region, CO2 is present. Okay, CO2 is present. CO2 is present. Now once again, you do the iodine test. And the result is very shocking now. Once you do the iodine test, the result is very shocking. The result tells you that this location right here, which is kept under KOH, there is no photosynthesis. No photosynthesis. Here, yes, photosynthesis occurred. Photosynthesis has occurred here. There is no photosynthesis in the KOH region. How do we know that? Because in this region there is no starch formation. In above region there is starch formation. Yes, starch is being formed. Blue-black is being formed. Starch is being formed. So what does it tell you? This experiment tells you, Moll's leaf experiment tells you that CO2 is essential for photosynthesis. It's written here, don't worry. The notes will be given to you, all of you. CO2 is essential for photosynthesis. Done. Clear? If the second experiment is clear to all of you, tell me green in the chat quickly. So all of you know my telegram, right? It's Basavaraj Sir Biology. Sir Biology. On this telegram channel, you will find all the notes, and also you will find the same notes also on Vedantu NEET English channel telegram. Every single detail, update, everything will be on that Vedantu NEET English telegram. Quickly everyone. Green in the chat if you understood the second experiment. Second experiment basically tells you that if KOH is present, no starch, no photosynthesis, tells you that no CO2, no photosynthesis. CO2 present, photosynthesis will happen and starch will be produced. Clear? Now, next experiment. Next experiment is by famous Joseph Priestley. Next experiment is by Joseph Priestley, which also called as bell jar experiment. It's also called as what? Bell jar experiment. Again, simple experiment, very simple experiment. Just look at the diagram. See here, we have a jar here. Inside the jar we have a candle and we have a mouse. Now inside this candle there is something very pure. Inside this jar there is something very pure. Something very pure inside the jar. Something, I'll tell you what is something. Wait. Something very pure is there. Now after some time what happens? Whatever something pure which is there inside, right? It has been completely used up. It has been completely used up by what? It has been completely used up by the burning candle and the respiration by the rat. So eventually the cute rat is dying, the candle also is extinguished, gone. So something was present there. Something pure was present there. It's being consumed. That something pure is being consumed by the burning candle and the rat. That something pure is nothing but oxygen. It is nothing but the oxygen. Oxygen. So the burning of candle and the respiration by the rat completely consumed the oxygen, and once the oxygen was completely consumed, the rat died, the candle also got extinguished. But once Joseph Priestley smartly kept a plant inside. It is a mint plant. He kept a mint plant inside. He kept a mint plant. Now what does this mint plant do? This mint plant is doing magic. What is the magic? Mint plant is restoring. It is restoring all the oxygen which is being consumed by the rat as well as the burning of candle. So all the oxygen is coming back. All the oxygen is again coming back. Yes, oxygen is there. Why? Because of the mint plant. Because of mint plant. Oxygen is coming. Because didn't we learn the starting? Didn't we learn the starting that plants will release oxygen as a byproduct? Yes. So if oxygen is there, will the candle continue to burn? Yes. Will the rat survive? Yes. Are they surviving? Yes. Rat is surviving and your candle is also burning. Candle is also burning. So what does it tell you, Mega? Mega, what does it tell you? It tells you that oxygen is important for photosynthesis. Done. That's it. That's it. This is Joseph Priestley experiment. He told you that plants restore the air. Whatever breathing animal and burning of candle removed. So oxygen was removed. Oxygen was removed by the burning of candle as well as your respiration by the rat. But plant did what? Plant did the magic. Plant completely restored the oxygen. Completely restored the oxygen. So there's one point need to remember. Oxygen was discovered. Oxygen discovery was done by Joseph Priestley in the 1774. In the year 1774. Done. Such an easy concept, students. Every single concept in this chapter is this easy. This easy. You do not have to break your head. Don't have to break your head. Okay. Now, next one is Jan Engine House. This guy is very funny. Okay. Jan Engine House is like, what do I say? Same but different. Same but different. So what do you do? Jan Engine House made the same experimental setup. He made the same experimental setup just like Joseph Priestley. Completely copied him. But he made small changes. What are the small change? The small change is that he did the experiment in the daytime as well as in the night time. That's all. In the daytime he observed bubbles were formed and he confirmed photosynthesis is happening. Because photosynthesis byproduct is what? The byproduct of your photosynthesis is oxygen. Yes. So in the daytime oxygen was evolved. Photosynthesis is happening. But at the same time, not same time, obviously night time. In the night time, no bubbles. No bubbles. It tells you what? In the night time, when there is no sunlight, when there is no sunlight available, photosynthesis will not happen. Photosynthesis will not happen. Now, what is the derivation from this experiment? What did we learn from this experiment, students? From this experiment right here, we learned that sunlight is important for photosynthesis. Simple. So, because night time, nothing is happening. See, sunlight is essential. Bubbles formed around the green plants. Only on the green plants, bubbles formed. Bubbles means what? Oxygen. Bubbles means oxygen. That was your Jan engine house. Okay. Clear? Green hearts in the chat quickly and smash the like button if you're enjoying the session. If you're able to understand every single point of the session, quickly mark your attendance and green hearts in the chat. Yes. Now, the next experiment. Not rather experiment, the discovery. Okay. It was by your Julius von Sauch. Julius von Sauch. He basically told you that all the food which is made in a plant is stored in the form of starch. All the glucose is stored in the form of starch. An entire process of photosynthesis is happening in the chloroplast. Provided evidence for production of glucose by plants. Plants are producing glucose. His studies show that chlorophyll is located inside the chloroplast. That's it. Chlorophyll is present where? Chlorophyll is present inside the chloroplast. Done. Leaves synthesize glucose, which is later stored as starch. 3 points, done. That's all. Nothing much. Only 3 points, very easy to remember. You don't need to read the entire big paragraph. 3 points, you're good to go. Done. Done, 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 done. The next one is by your T.W. Engelmann experiment. Not written here, okay. T.W. Engelmann experiment. Now students, I want you to focus now. I want you to focus extremely high now because I'll be teaching you this experiment and we'll be doing it. As I'm not doing it actually, but I'll be showing you the experiment. Okay. Ready all of you? All of you ready? Quickly in the chat. So this experiment is very, very simple. Very simple. So T.W. Engelmann worked on the light. So what did he do? So he took a prism. Yes, there is a prism. He took a white light. So when you pass the white light through a prism, obviously there is scattering of light. Right? Obviously there will be complete scattering of light. So once there is scattering of light, your T.W. Engelmann did a very smart move. At the end of this, he kept a complete test tube of a particular algae. He kept what? He kept an algae here. Okay, fine. He kept an algae. The name of this algae is called as Cladophora. The name of this algae is called as Cladophora. Cladophora algae. Okay, Cladophora algae. Clear? Now in the same test tube, this is your test tube. This is your test tube. In the same test tube, he also added certain aerobic bacteria. He also added what? He also added aerobic bacteria there. Okay, students. So we have your green algae, Cladophora, and we also have what? We also have the aerobic bacteria inside. Aerobic bacteria inside. Done. Aerobic bacteria is there. Gopika ma'am in the chat. Hello, Gopika ma'am. Okay. So we have the test tube. Inside the test tube, we have the algae called as Cladophora, and we also have the aerobic bacteria. Done. Done. Till here, in doubt? Till here, I believe there should be no doubt because experiment is very interesting. So when you pass white light through a prism, and once there is scattering of light, obviously different light will fall on. Scattering of light is happening? Yes, sir. Scattering of light is happening. T.W. Engelmann noticed that. T.W. Engelmann noticed that at this region right here, can you see this region? And this region. These two regions, he noticed that this aerobic bacteria is clustering here. Nicely it is clustering. One gang it is making, a nice gang. Can you see? In these two regions, aerobic bacteria is making a gang. They are playing chain here. Can you see? Nicely they are making one big gang here. In these two regions. Then he noticed in these two regions, why were there more aerobic bacteria? Now, reason it is called as aerobic bacteria. The reason it's called as aerobic bacteria because it requires what? It requires oxygen. It will take oxygen. So, technically speaking, in these two regions, there was more O2. Can I tell? More oxygen. Yes, sir. Since in these two regions, if more aerobic bacteria is growing, there is more oxygen in these two regions. Okay, more oxygen in these two regions. And so if there is more oxygen, can I tell in these two regions photosynthesis is also higher? photosynthesis is also higher in these two regions. Yes. If oxygen is released more, if the byproduct is only more here, it means photosynthesis is also higher. Yes. And later we got to know, not later, that time only we got to know that this region right here is the red region, where red light is falling, and this is the region where we have the blue light. So what is it? What do you understand from this experiment, students? From this experiment right here, we understand that in the red light region as well as your blue light regions, in the red region and blue regions of a spectrum of light, more photosynthesis takes place. Photosynthesis is higher. The rate of photosynthesis is higher in the red region as well as the blue region of your light spectrum. Beautiful. That's it. Did all of you understand? It's very simple. If you read it in NCERT, you might get confused, but with the help of diagram, it becomes very, very, very easy to understand. See here, more aerobic bacteria means more oxygen. More oxygen means more photosynthesis in the blue region. Similar case in the case of your red region, more oxygen, more photosynthesis. Done. Green as in the chat if you understood. Now students, one more point to be added here. This right here, student, this right here is actually the first action spectrum. It is the first action spectrum. First action spectrum right here. Now what is the action spectrum? We'll discuss detailing in later on. Don't worry. It's very easy to understand. See here, what do we understand? Conclusion: aerobic bacteria detects the presence of O2. Yes. Aerobic bacteria will do what? Presence of O2. Done. Formation of O2 means what? Higher where? Higher in red and blue light. Yes. Higher in red and blue light. Now blue and light wavelength is optimum for photosynthesis. Blue and red light is optimum for photosynthesis. Boom. Done. One more experiment done. Students, this chapter is very easy. If you understand this chapter, if you try to mug up this chapter, it can get a little difficult. You can mug up, but it is a very bad thing to do. But understand the chapter, you don't need to mug up at all. Okay. The next thing is okay, before we go to the next experiment, did we learn that carbon dioxide is important for photosynthesis? Did we learn that? Yes, sir, we learned that. Did we learn water is important till now? No. We didn't learn, right? We didn't learn. Did we learn sunlight is more important? Yes. Remember, same but different we learned, right? Jan engine house. Chlorophyll is more important? Yes. Starting variegated leaf experiment. CO2, KOH experiment. And we learnt, Von Soch told us that glucose is produced. Yes, glucose is produced. Did we learn oxygen is produced by Joseph Priestley? Yes. Joseph Priestley also we learnt. So, we are actually covering your entire reaction of your photosynthesis with the help of experiments. With the help of certain experiments, students. With the help of certain experiments. Beautiful, right? Very easy chapter, right? Now, the only thing which is missing right now here is water. H2O, water. Okay, H2O, water. For that, we have one more experiment called as your Corneal-Van experiment. Cornelius-Van experiment. Sounds very scary, but it's very stupid and very easy. Very easy. Because Cornelius was not a botanist. He was a microbiologist. He was a microbiologist who worked on botany. Yes, he worked on certain plants also. So his main work was microbiology. If he is a microbiologist, he'll obviously work on microbes. So his main work was on green and purple sulfur bacteria. His main work was on green as well as purple sulfur bacteria. When he worked on these two bacteria, he noticed that when the green and purple sulfur bacteria are doing photosynthesis, they are also doing photosynthesis, but they are not producing oxygen. No, oxygen is not released as a byproduct. No. In fact, H2 is produced. Sorry, not H2, sorry. Why am I saying? Sulfur is produced. Sulfur is produced as a byproduct. So this made us realize that the hydrogen donor in the case of your purple and green sulfur bacteria is H2S. it is H2S, which gets converted to H plus, SO2 minus. The splitting here. But in the case of plants, students, in the case of plants, it is water is being split. Yes, oxygen as a byproduct, and we also have the H plus, which is taken up by the plants. Did we learn that in the starting? Did we learn in the starting the plant cell which I drew for you 30 minutes back? Did we learn there that the water, H2O, hydrogen is sent to glucose making? If you don't remember, let me show it to you, because some of you are very forgetful. Some of you are very forgetful. See here. In the starting of this class, we learned here, right here. See here. Water is H is taken to the glucose and oxygen is the byproduct here. Oxygen is the byproduct here. Very easy chapter, students. Now the next thing which you need to do is solve some questions. Ready? So what did he do? Cornelius Van Til told us that oxidation of water results in the liberation of O2. We learned that. Water provides the hydrogen for the reduction. See, reducing CO2 to carbohydrate. We learned all these words. Reduction, CO2 is getting reduced to glucose. Water is undergoing oxidation. So oxygen evolved from the green plants comes from the water, not from CO2. If you all of you understood quickly in the chat, green has in the chat, and smash the like button, all of you. Clear? Can you solve some questions now? Simple question on your board. Go. I'll give you 10 seconds to answer this question. 10 seconds. 10 seconds. Photosynthesis is categorized as the physico-chemical process. Why? Answer is very simple. It is a physico-chemical process because light energy is getting converted to chemical energy. Light energy is utilized to synthesize your organic compounds. That is your chemical energy. Right? Nuclear, it cannot be. Electrical, it cannot be. Organic compounds utilized to obtain light energy. No, light energy cannot be obtained. So, organic compounds are made. Simple. Which of the following organisms do not release oxygen as its byproduct during photosynthesis? We learned this around 2 minutes back, so please answer this very quickly. 2 minutes back we learned it. 2 minutes back. Legit 2 minutes back we learned it. Cyanobacteria, purple and green sulfur bacteria, both A and B. None of this. Answer is purple and green sulfur bacteria. Okay, done. Now, can we go to the next concept, all of you? Can we go to the next concept, beautiful concept called as site of photosynthesis? Site of photosynthesis, which is called as a chloroplast. But before we go to the chloroplast, can we understand where does the photosynthesis happen? Now, if you are walking on the road, if all of you are walking on the road, if a kid asks you, if a kid will ask you, Anna, Akka, if this photosynthesis, where does it happen? If you tell to that kid chloroplast, will he understand? No. So all of you will tell what? All of you will tell, obviously, you will tell it happens in green plants. It happens in green plants. The kid will be happy. The kid will be happily, he'll be like, okay, fine, green plants, it happens, photosynthesis. Now, if a class 10 student asks you, class 10 student or class 10 student asks you, where does the photosynthesis happen? If you tell green plants, he'll be like, even I know that. Tell me more. Yeah, tell me more. So, within the green plants, there's something called as mesophyll cells. Yes, there's something called as, in green plants, we have leaves. Obviously, it happens in leaves. We have something called as mesophyll cells. Mesophyll cells. It happens in the mesophyll cells. But if I ask you, where does the photosynthesis takes place? You should be able to tell me, sir, photosynthesis is the process which takes place inside the chloroplast. It happens inside the chloroplast. Done. That is the first answer. It happens inside the chloroplast. Now, can we draw the chloroplast? Yes, we can. So, quickly all of you open your books. Quickly all of you open your books right now, because we will be drawing the chloroplast structure, the double membrane structure. Did you join there? I didn't join. You know what? I'll just shape. Students, this is your chloroplast. This is your chloroplast. So your chloroplast has how many membranes? Since it's a double membrane structure, just like your Golgi, just like your mitochondria and nucleus, double membrane structure. So it will have a outer membrane and an inner membrane. Yes. So this right here is the outer membrane. Okay, sir, we have the outer membrane. Then we also have the inner membrane. We also have the inner membrane right here. Inner membrane. Done. Outer membrane as well as your inner membrane. Done. Now if you look inside the chloroplast, we have certain coin-like structure. Inside the chloroplast, we have certain coin-like structure. This, can you see? We have certain coin-like structure. These coin-like structures are called as thylakoid. They are called as what? Thylakoid. Right? They are called as your thylakoid. Now, this thylakoid, it will never be present as single. It will be in a big group. So when we have a lot of coin-like structures and they are present in a stack, the entire stack is called as grana. Grana or granum. Grana or granum. Grana or granum. Okay? Grana or granum. Okay? So we do not have only one grana. In fact, inside the chloroplast, we have many granas. We have many granas. Many granas. Now each individual grana, students, each stack of coins grana is interconnected. It is always interconnected. The connection is called a stroma lamella. Let me write here. Once again, change the color. Here. Clear? This is your stroma lemma, right? Stromal lemma. That is your connection. Now, what is this matrix? Do you see this matrix here inside? Yes, sir. Everything will have a matrix. So we have a matrix here. We have a matrix also. We have a matrix. This entire matrix is called as what? Stroma. Entire matrix is called as stroma. It's called as stroma. So we have stroma, matrix, we have outer membrane, inner membrane, we have coin-like structure called as thylakoid, and entire stack is called as grana. Interconnecting is called as stromal lemma. Okay, stromal lemma. Now this here I can stop. Obviously it will have certain ribosomes. Let me draw it. Sir, you will like, no sir, you didn't draw the ribosomes only. Don't cry. We have the ribosomes here. We have the ribosomes. 70S ribosome. Happy? 70S ribosomes. Now apart from this we have oil droplets, starch content, that you can draw on your own. Starch content, oil droplets. That is also there. Okay. Now apart from this you should understand few things here which is actually more important. That is going to be the something called as photosystem. Technically you will be studying about photosystem later on, but right now you need to understand what is the photosystem, where it is present. On this stroma, on this thylakoid surface, we have two different photosystems. We have PS1, PS2 as well as, one second, as well as PS1. That is photosystem 2 and photosystem 1 are present on the thylakoid membrane or on the grana. On the grana or thylakoid we have PS2 and PS1. But on the stromal lemma, can you see this one, the connection bridge? On the bridge do we have photosystem? Let me draw it pink color, easy to understand. We have two here. Okay, we have two here. On this bridge also we only have PS1, but we only have PS1 on the bridge. On the stromal lemma we only have PS1, but on the thylakoid we have both PS2 and PS1, both are there. Students, this concept is very important because we will be discussing this when we come to photosystem, and I'll be telling you what is photosystem. You don't have to by heart anything. Okay, easy all of you? Did all of you understand? So if you understood this, can I add one more point here? Yes sir, definitely you can add one more point. The other point which I have to add here is very simple again, very simple point. That is, in the stroma we have the dark reaction. we have the dark reaction in the stroma. So dark reaction in the stroma, and in the thylakoid or the grana we have the light reaction taking place. Light reaction taking place here. Light reaction taking place. Done. If all of you understood this, quickly green us in the chat, because it is very important to confirm if you're understanding or if you're just blindly sitting there. It's important to confirm, right? So this is your entire structure of your chloroplast. Outer membrane, inner membrane, matrix is called stroma, where dark reaction takes place. We have ribosomes. They have coin-like structure called as thylakoid. Entire stack is called as grana. On the thylakoid, we have light reaction. The connecting bridge between the two thylakoids or to the grana is called as stromal lemma. In stromal lemma, we have the PS1, only PS1. But on the grana thylakoid, we have both PS2 and PS1, both are present. Done. If this you understood, you're halfway into the light reaction, dark reaction. Halfway there. Halfway there already. You're halfway there already. Okay. Now, just revise. Let's see what we just learned. See here. This is your entire chloroplast structure. Stroma, matrix, stroma lamellae. Each one is called as thylakoid. Entire stack is called as grana. Stroma lamellae is nothing but your connecting bridge. That's all. That's all. Now, see this question, all of you. See this question quickly. See this question. Which of the following photosystem is predominantly present in stroma lemma or stromal lemma or stromal lamella? Same meaning. Okay. Stromal lemma or stroma lamellae. Which is present? We have PS1, PS2, both PS1 and PS2, none of the above. What did we just see? We saw that on stroma lemma we only have the PS1. So what is the answer? The answer is going to be only PS1. Done. So when you actually understand the concept, right, this type of questions becomes very easy to solve. Very, very easy to solve. Okay, on the stromal lamellae. Now, can we go to the pigments now? Can we go to the pigments? Can we start with the pigments? What are the different pigments present in the photosynthesis? For if I ask you, sir, tell me some 3 pigments, all of you, quickly in the chat. Some 3 pigments you tell me. 3 pigments. You like chlorophyll, 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 chlorophyll. Chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D. Smart, no? But no. It doesn't work like that, because here we will be learning about chlorophyll A, chlorophyll B, xanthophyll and carotenoids, as well as carotenoids. Okay. But how do you extract these pigments? Is there any technique to extract the pigments? Yes. What is the technique called as? Chutney making. It is called as chutney making. How do you make the chutney? All of you. Take a mortar and pestle. Take a mortar and pestle. Take a leaf. Take not one leaf, don't be confused. Take around 5 to 6 different leaves. Take the leaf and you add it to the mortar and pestle and nicely grind it. Nicely grind it. When you're making the chutney, do you add water? When you're making coconut chutney, you add water. No, some water you add. So here also you need to add some amount of water. But you don't add water. Actually here, you add a separate solvent. So that everything... So see, the reason you add solvent is that because the outer layer of your leaf has the cellulose, right? If the cellulose has to break down, water is not sufficient. For that you need to add solvent like acetone. So once you add acetone or any other solvent, the entire cell wall will break down easily and all the pigments will come out. Extra information I gave you. Then you add it to a strain and you bring down the liquid. Okay, you bring down the liquid. Now you take this liquid and you take one small straw. With the help of a small straw or a small capillary tube. Not straw, we call it capillary tube. With the help of capillary tube, you add one dot here and wait for 2 seconds. Wait for 5 seconds. Wait for 10 seconds. Take one more drop, add one more drop. Then take one more drop, add one more drop. So once you do that, we have a very good concentration. We have a very good concentration of your extract here. Nice concentration of extract. Now you take this leaf and you add this leaf into one more solvent. Test tube, solvent is there. Now as the solvent will move up, as the solvent is trying to move up against gravity, right? It will take the pigment along with it and it will bring the pigment up. It will technically separate the pigment based on the retention time and retention property. Okay, so your pigments are getting separated now. Pigments are getting separated: chlorophyll A, chlorophyll B, xanthophyll, carotenoids. And this technique is called as paper chromatography. It's called as what? Paper chromatography. Paper chromatography. Okay, paper chromatography. Now, with the help of this paper chromatography, you will be able to find chlorophyll A, chlorophyll B, xanthophylls, as well as carotenoids. Carotenoids. But if I ask you, students, right now, what is pigment? You will tell me, sir, pigment is a color, right? All of you will tell me, sir, pigment means color. You will tell me that? Yes, pigment means color. But you're half correct. Technically, you're correct only, but what exactly is a pigment? That is very important to understand. Pigment is actually a chemical compound. Pigment is actually a chemical compound. Let me write here. Pigment is actually a chemical compound. What does it do? It basically converts. It converts light energy. It traps the light energy, not converts, it traps the light energy. Traps or converts the light energy into chemical energy. Into chemical energy. That is the work of a pigment. That is the work of your pigment. Convert light energy into chemical energy. I'll give you a small proof. Small proof I'll give you. See this. This is your pigment. Right? This right here is your pigment. Okay. Let me write green only. You're like, sir, different color. Make it green. This right here is your pigment. Right, is your pigment. Now, on this pigment, if there is a incident, photons are coming, incident light is coming, okay, light is falling on this pigment. Now, what will happen? In this pigment, are there electrons? Yes. So, from this pigment, electrons are being excited. Electrons are being excited. And this electrons will get excited only at a fixed wavelength. That is very important. Fixed wavelength. At a fixed wavelength, all the electrons will get excited. Yes, electrons will get excited. Now, if the electron is excited, chemistry, basic chemistry, if it goes to higher orbitals, will the electron come back? Like, yes, sir. If the electron has gone up, it will also come down to the ground state. The electron will also come back to the ground state. Now, student, when the electron is coming back to the ground state, when the electron is coming to the ground state, there are few possibilities of events happening. What is the possibility? The first possibility is very simple. That is, when the electron is traveling back, can it emit some light? It can emit light. Yes, sir, it can emit light. It can emit energy. Yes, it can emit energy. Basic chemistry. Yes, sir. When it is coming down, it can also transfer the energy. Transfer the energy to another pigment. Yes, it can transfer energy to another pigment. That is also possibility. When the electron is coming down, it can transfer the energy to another pigment. Or it can use the energy. It can use the energy to do some work. Now, what is this work? That work is nothing but photolysis. Photochemical reaction. It's called as photochemical reactions. Photochemical reaction. Do you realize what we just learned? We learned chemistry to understand biology. That is the beauty of this chapter. We just understood a little of chemistry to understand biology, because these two we are not interested. These 2 points we are not interested, because that you will be studying in your chemistry. What we are interested right now here is these 2 points. Why? because these 2 points we'll be using it to understand. We'll use these points, 2 points, to understand what is the meaning of axillary pigment, what is the meaning of reaction center, what is the meaning of light harvesting complex, what is the meaning of photosystem. You might have by-hearted all of this. You might have heard about photosystem, but today you will be learning what exactly is photosystem. Ready? Ready, all of you? So I believe all of you understood these points, right? So when electron is going to higher orbital, it is coming back to the ground state. When it is coming back to the ground state, it can emit light, emit energy, transfer the energy to another pigment, use the energy to do work. That is called as photochemical reaction. That is called as photochemical reaction. So if you understood these 2 points, we will be using these 2 points to understand biological concepts now. Biological concepts. For example, before we have to teach you some other concept. I don't know why this is here. We will understand that. Before that, can we quickly understand what is the meaning of action spectrum and absorption spectrum? Very small concept, very easy concept, very easy concept. Can we quickly understand? It sounds very difficult, right? Absorption spectrum, action spectrum. Very easy it is. Your absorption spectrum is a spectrum. Listen to my golden words right now. Your absorption spectrum is a spectrum which tells us that at which wavelength which pigment is getting absorbed. That's it. Let me repeat for you. Your absorption spectrum tells you that at which wavelength, 400, 500, 600, 700, at which wavelength which pigment is getting absorbed. That's all is the absorption spectrum. That's all. For example, your chlorophyll A is getting absorbed around 450, chlorophyll B at 480, carotenoids around 500, xanthophylls will somewhere here, right? So every single pigment, not here, it cannot be here, sorry. Everything is below 680, here, somewhere here only, right? So every single pigment has a specific wavelength where it can get absorbed. That is absorption spectrum. That is absorption spectrum. Did all of you understand? Menons, I wish I could talk in Hindi, but very difficult. I am not very proficient. I cannot speak very well. And if I speak, you will not understand anything. That is a bigger problem. If I speak in Hindi, you will not understand anything. It is that bad. All of you understood, right? So absorption spectrum is a spectrum at which it shows you that which pigment is getting absorbed at which wavelength. That's it. Now, all of the points are given here, students. All the points are given here. You can read the points. It will be easy to understand. Okay. Now, what is the action spectrum? What is action spectrum? Okay. Now, action spectrum tells you what? Action means some work. Action means some work is happening. Now, what work is happening, students, here? The work which we are talking about here is photosynthesis. Photosynthesis. So, your action spectrum, students, write down these words. Your action spectrum tells you at which wavelength photosynthesis is happening. That's all. At which particular wavelength photosynthesis is happening. For example, at these 2 wavelengths, blue and red, at wherever the blue light is there and wherever red light is there, at these 2 wavelengths, photosynthesis is higher. Photosynthesis is higher. Didn't we learn this somewhere around 30 minutes back? Didn't we learn this somewhere, students? Didn't we learn it somewhere? Didn't we learn it? All of you forgot, huh? Nice. Didn't we learn it in your TW Engelman experiment? At one particular wavelength, red and blue regions, photosynthesis is happening more. Action is happening more. That's why I wrote here action spectrum. Action spectrum. Done. That is your action spectrum. That's all. At which wavelength photosynthesis is happening. Photosynthesis is happening. Easy to understand, students. Very easy to understand. Very easy to understand. Action spectrum shows the effectiveness of different wavelength of light for a particular reaction. Obviously, different wavelength of light has different reactions. Then shows which wavelength of light is most effective utilized in a specific chemical reaction. The blue light and the red light are obviously more superior. They're able to do photosynthesis much more better. That is your action spectrum. Done. Then all of you, quickly, in the chat tell me. Say this is your absorption spectrum from your NCERT, which is telling me that at this particular range, absorption is happening of this pigments, absorption spectrum. This is your action spectrum. That is telling me at this region right here, at this region right here, and this region right here, action is high. That is, photosynthesis is very high here. Photosynthesis is very, very high here. Done. Now, your NCERT has one more diagram, one more table. In fact, if you have noticed this, we have one more table here. Now, this is little difficult to understand if you are not aware of what specifically they are asking you here. First of all, let me tell you, this particular table, this particular graph, not table, this particular graph is actually the baby of these two graphs. So take this graph, and you superimpose them. This graph, you superimpose them, and you end up getting this one. Yes. And students, let me tell you one point which will help you to understand this better. This graph right here, if you read the legend of this, that is very important. If you read the legend right here, it says graph showing action spectrum of photosynthesis superimposed on the absorption spectrum of chlorophyll A. This is only and only for what? This is only and only for chlorophyll A. That is very important to understand. Okay. Now, I will open your eyes little now. I'll open your eyes and I'll teach you some extra concept here. Listen very carefully. Listen very carefully. This light blue is what? Light blue is nothing but your absorption. Okay, sir. At this point right here and this point right here, absorption is very high. Okay. Absorption is very high. Absorption is very high. If light is being absorbed at these two wavelengths, photosynthesis will happen or not? Tell me in the chat. At these two regions, if absorption is very high, these two regions, absorption is very high, will the photosynthesis also happen very high in that locations? Yes. Obviously, plant is taking light, plant will do work. So, can you see? At those two locations itself, your rate of photosynthesis is also very high. Rate of photosynthesis is also very high at these two locations because absorption of light is also high. Yes, since absorption is high, photosynthesis is high. But, what is the but here? If you notice, at these regions right here, this till from here to here, there is less absorption. There is very less absorption, right? Very less absorption. Very less absorption. If there is no absorption in this range, why do I see some amount of photosynthesis here? See here, if you notice in these regions, very low absorption. But even though there is very low absorption, photosynthesis is still happening. How and why? The answer is very simple. In your school, we have one principal. Even when the principal is absent, will the classes go on in your college or school? Tell me. Even though principal is absent in your school, will the classes continue to go on? Yes, classes will continue. Why? Because even though principal is absent, are there class teachers in your class? Teachers are coming. Yes, teachers are coming and they're making sure the classes are happening. Similarly, even though chlorophyll A is not absorbed, here, there is something called as accessory pigments. These accessory pigments such as chlorophyll B, xanthophyll, and carotenoids, as well as carotenoids, they are getting absorbed. They are getting absorbed. Since carotenoids, xanthophyll, and chlorophyll be getting absorbed, they're absorbing the light. They're absorbing the light, they're absorbing the energy, and they're giving the energy to chlorophyll A. They're giving the energy to chlorophyll A. Even though chlorophyll A might not be absorbed here, but axillary pigments are getting, they're absorbing the energy and they're sending the energy to chlorophyll A. Chlorophyll A. So what is the game here? The game here is the teachers. Who are the teachers? The teachers here are the accessory pigments. The teachers here are the accessory pigments. So do you notice? So even though there is no absorption, photosynthesis will still happen. Photosynthesis will still happen. That's all. That's all. Did all of you understand? Tell me in the chat, did you all of you understand the presence of other pigments? Yes, no, yes, no. Done. This is very important graph to understand. Very important graph to understand for all of you. See the question here. At which wavelength of color of light maximum photosynthesis in plants is observed? Very easy question. 10 seconds, go. Time starts now. Very easy question. Time starts now. Yes, absorbed light and energy is from the auxiliary pigments is sent to the chlorophyll A. Done. Blue and red regions, absorption is maximum. Absorption is maximum. Now students, we spoke about pigments. We spoke so much about pigments. Can we classify the pigments now? Can we understand more about pigments? Different types of pigments? Yes sir. Students, there are two types of pigments. The first type of pigment is called as your chief pigment. Chief pigment. Chief pigment is like a principal. Is like a principal. Main boss. Principal. Then we also have certain accessory pigments. We also have certain accessory pigments. They are like the teachers. Teachers. Under your chief pigment, we have only and only chlorophyll A. Only and only chlorophyll A. Under your auxiliary pigments, whom do we have? Chlorophyll B, carotenoids, and xanthophyll as well. We have xanthophyll as well. Now students, do you notice there's something written here? Blue, green, yellow to orange, yellow. Is this important to study? Is this important to study? They're giving you the color of the pigment. Is the color of the pigment important to study? You might think, sir, very trivial, very simple. But students, this simple thing right here is a NEET PYQ. This simple thing right here is a NEET previous year question. That's right. This right here, simple. Color of a pigment they have asked you. Color of a pigment they have asked you in your examination. So, you need to remember this. You need to remember this. Do you need a small trick? Do you need a small trick to remember this? Vib. G-Y-O-R. What is this VibGyor, students? What is VibGyor? V-I-B-G-Y-O-R. VibGyor is nothing but the colors in the rainbow. Colors in the rainbow. VibGyor. All of you know VibGyor, right? VibGyor. See this? Blue, green, chlorophyll A. Chlorophyll A. Green, yellow, green, chlorophyll B. Chlorophyll B. Then we have yellow to yellow orange. Yellow to orange is carotenoids. 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 And then we have yellow, just yellow. We have xanthophyll. We have the xanthophyll. Done. That's it. Very easy to remember. Very easy to remember. VibGyor. Blue, green, chlorophyll A. Green to yellow, chlorophyll B. Yellow to orange, carotenoids. Yellow is xanthophyll. Yellow is xanthophyll. Simple. Now students, the next thing you need to understand is how do they work? How do they work? Before that, let me teach you one more concept. Before they understand how do they work, we learnt about auxiliary pigments and we learnt about your chief pigment. Now when this auxiliary pigments and chief pigment, when they are present together, in a packet like structure, that is nothing but your light harvesting complex, LHC. So what is LHC? LHC is nothing but antenna or antenna is what? Antenna is your axillary pigments. And reaction center is what? Reaction center is your chief pigment. Chief pigment. Chief pigment. So when your chief pigment plus your axillary pigments together, when they're present, that's called as LHC. It's called as light harvesting complex. So pigments are organized. Pigments, your reaction pigments as well as your axillary pigments, reaction as well as your chief pigment are arranged together, organized into discrete photochemical light harvesting complexes, LHC, within the photosystem. Again, very importantly, I'll get back to this. I'll get back to this line. Within the photosystem. So do all of you understand what is light harvesting complex now? Light harvesting complexes are the pigments which harvest light. Which harvest light. Okay. LHC are made up of hundreds of pigments, molecule bound to proteins. We have hundreds of pigments. We have antenna pigments and we have reaction centers as well as your reaction centers. Now, let me get the concept even more clear now. Can we get the concept even more clear now? Yes. For example, students, this is your concept should be clear. This is your chlorophyll A. Imagine this is your chlorophyll A. It works at around 680 nanometer. 680 nanometer. Okay. Then we have around, let's take this as chlorophyll B. Let's take it as chlorophyll B. Chlorophyll B works at around 600 nanometer. Take it as 600 nanometer. Okay, 600 nanometer. Chlorophyll B. Then we also have xanthophyll. One second. We have xanthophyll. Xanthophyll works at around what? 550 to 600 nanometer. 600 nanometer. Then we have what? Then we have carotenoids. Then we have your carotenoids whose main range is around 500 to 550. Whose main range is from 500 to 550. I hope you're able to see this. Now what happens is sunlight is coming. Okay, sunlight is coming. The sunlight is, in the sunlight we have something called as PAR. PAR. Photosynthetically active radiation. Photosynthetically active radiation, which is mainly between 400 to 700 nanometer. 700 nanometer. Photosynthetically active radiation. Now, whenever we have wavelength of 500 wavelength of light, absorption is done. Absorption is done. Done. It's absorbing. We have 550 wavelength of light. It's absorbing the wavelength of light. Absorbing wavelength of light around 600. Whenever there is wavelength of 680, chlorophyll A is absorbing the light. It is absorbing the light. Absorbing the light. Okay. It is absorbing the light. Okay. Absorption is done. Now, one thing you need to remember here is all of these carotenoids, xanthophyll and chlorophyll B, they are called as your accessory pigments. They are called as what? Accessory pigments, also called as your antenna. They are called as antenna. Now, why are they called as antenna? Because they will trap the sunlight. They will trap the light at this wavelength. They will trap the light at this wavelength, and they will only thing they'll do is transfer the energy. Transfer the energy to the chlorophyll A. Chlorophyll A. All the energy is sent to the chlorophyll A. Clear? So the main work of your antenna, is to trap the light at different wavelengths and take all the light, harvest all the light and send it to the main reaction center. Main reaction center. That's why it's called as what? That is why it's called as what? Light harvesting complex. It is harvesting the light at different wavelength and it is sending it to the chlorophyll A. Now, why are they sending to chlorophyll A? Because chlorophyll A has the enzymatic property. Your axillary pigments do not have enzymatic property. They do not have enzymes. They do not have the enzymes. The enzymes are only present in the case of your chlorophyll A, the main boss. Chlorophyll A, the main boss. Clear? I believe this concept should be very clear to all of you. Very clear concept. That's why they are called as light harvesting complexes. Light harvesting complexes. Beautiful. This is your reaction center, which is chlorophyll A, and then we have antenna. See, can you see here? Now, okay, fine. That is light harvesting complex. That is light harvesting complex, LHC. Then what is a photosystem? What is a photosystem? Are photosystem light harvesting complex same to same or the different concept? No. You can mug up all this. But you need to understand this. They're actually very different. So before I tell you photosystem, listen to me very carefully. What is a photosystem? Photosystem is actually made of three things. Photosystem is actually made up of three different things. This is your photosystem. Photosystem is made up of light harvesting complexes, which has your axillary pigments, also called as your antenna, also has your reaction center. Okay, it also has reaction center. Apart from that, we also have certain electron carriers. Apart from electron carriers, we have a lot of enzymes. Electron carriers such as your pheophytin, which we learn in the non-cyclic photophosphorylation, FES, ferredoxin, that FES, iron sulfur centers. Are also type of your electron carriers. So when we have your electron carriers and light harvesting complex as well as enzymes together they are called as photosystem. Clear all of you? Together they are called as photosystem. That's why here we wrote a line. Can you see? Pigments of organized into discrete photochemical light harvesting complexes within the photosystem. LHC are present where? LHC are present within the photosystem. Beautiful, right? Concept understood, all of you? Concept should be understood. Okay? Tell me yes or no in the chat. If all of you understood. Light harvesting complex. They will harvest the light. See here? It's called as photosystem. Now, location of photosystem. For example, photosystem 2 is located on the stacked regions. What is the meaning of stacked regions? Stacked regions are nothing but your thylakoid grana. Yes. Didn't we learn in the starting? This is your grana. On the grana we have what? On the grana we have the both PS2 and PS1. But on the stromal lamellae, what do we have? This is connection. This is stromal lamellae. On top of stromal lamellae, we only have PS1. Not PlayStation, photosystem 1. Photosystem. We know what is photosystem now. Okay. So, photosystem 2 is located on the stacked regions or oppressed regions of the thylakoid membrane. Done. Photosystem 1 and ATP synthase are found on unstacked regions, oppressed regions, protruding into the stroma. They are present here also. That's all. What is the difference? Difference is very simple. Photosystem 1 works at 7 nanometer. Photosystem 1 works at 700 nanometer. And your photosystem 2 works at 680 nanometer. Okay. Found both in grana and stroma lamellae. That is photosystem 1. For photosystem 2 is only found in your grana lamellae. This is grana lamellae. This is grana, right? This is grana. Grana lamellae, only photosystem 2 is found. That's all. Difference between photosystem 1 and difference between photosystem 2. 1 and 2. Now, can we start with the light reactions now? Light reaction one by one, we'll start now. Light reaction. Now, if I ask you what exactly is present in light reaction, if you look at your entire photosynthesis, it is divided into two parts. The first part is called as light reaction. The other part is called as your dark reaction. The dark reaction is also called as light independent reaction. The reason it's called as dark reaction because it happens in dark? No. The reason it's called as a dark reaction it is because it doesn't require light. It doesn't require light. It doesn't require light. Okay? Doesn't require light. It occurs within the membrane system of chloroplast. Yes, that is on the thylakoids. We have this. While your dark reaction happens in the stroma. Happens in your stroma. And in a light reaction, there is trapping of light with the help of LHC, which is the part of your photosystem. Yes, photosystem 211. That is trapping of light, and synthesis of ATP and NADPH is produced here. ATP and NADPH is produced in your light reaction. The same ATP and NADPH is used in your dark reaction. Same ATP and NADPH is completely utilized in your dark reaction. When ATP and NADPH are utilized in your dark reaction, sugar is formed. Sugar is formed. Okay? Sugar is formed. That is the gist of your light and dark reaction. Light and dark reaction. Now we will go to the depths. We will go into the depths of your light reaction. Your entire light reaction can be divided into two categories. Entire light reaction can be divided into two categories. The first one is called as non-cyclic photophosphorylation. Why is it called as photophosphorylation? Photo means light, phosphorylation is ATP production. You are using light to produce ATP here in a non-cyclic manner. Here in this case right here, we are using light to produce ATP in a cyclic manner. It's a cycle. So can we start with your non-cyclic photophosphorylation now, students? Can we start now? Quickly in the chat, students, don't worry, the entire notes will be available on Telegram channel. Basavaraj Sir Biology, you can find the entire notes. Now I want you to do one thing. Open your book right now in front of me. Open your book and start writing with me. All of you. All of you quickly start writing with me so you can understand it better and point to point you can follow with me. Okay? Ready all of you? I want everyone to write with me now. Tell me if you're ready. Let's start. I'll drink some water. I'll drink some water and we can start with your non-cyclic photophosphorylation. Clear? Can we start now? Now let's start with your non-cyclic. Okay. The first thing you need to remember here is, let me draw it for you. One by one we can go. One by one we can go. Here we have the, space is enough, right? Yeah. Here we have your chlorophyll A. Chlorophyll A. Okay. Now chlorophyll A, is it a reaction center or is it auxiliary? You will tell me, sir. Chlorophyll A is the reaction center. The reaction center will have auxiliary pigments. Yes. There are auxiliary pigments attached to this. Auxiliary pigments are here. All the auxiliary pigments will send light. Yes, obviously will send light. Obviously will send light. So we have sunlight here. Sunlight is falling. Yes, sunlight is falling. Sunlight is falling. So once the sunlight is there, all the energy will be light harvesting complex. Everything is sent to chlorophyll A. Everything is sent to chlorophyll A. Because they do not have enzymatic activity, auxiliary pigments or antenna will send all the energy to chlorophyll A. Now this chlorophyll A works at your wavelength around 680 nanometer. 680 nanometer, right? 680 nanometer. And this particular wavelength, 680 nanometer. And this is called as your photosystem 2. Photosystem 2. Photosystem 2, 680 nanometer. Light is coming, falling on the reaction center. Now what happens? As soon as the light falls, electron is being excited. Yes. Electron is being excited. Okay, electron is being excited. Now, let me write for you. Two electrons are being excited. Electrons are being excited. Now, didn't we learn somewhere when electrons are excited, when they're coming down, they can transfer the energy to another pigment? We learned somewhere. We will use that here only. We'll do that here only. We'll use it somewhere here. Now from here, electron is taken up by. From here, electron is taken up by your primary electron acceptor. PEA. Primary electron acceptor. The primary electron acceptor here is nothing but your pheophytin. Is your pheophytin. All of you can see it, right? Pheophytin. Now from electron from pheophytin is sent to the next electron carrier. The next electron carrier is going to be plastoquinone. Plastoquinone to cytochrome B6F complex. Then it is given to PC, that is plastocyanin. Should I write it somewhere? Yes, I'll write here in the corner. So that you can always come back to this and you can easily remember this also. What do we have? PQ. PQ is nothing but your plastoquinone. We have CY, that is cytochrome. Why is it changing? Cytochrome B6F complex. B6F complex. Then we also have your PC. That is plastocyanin. Plastocyanin. Yes, plastocyanin. Now from here, electron is being transferred. Okay, electrons are being transferred. Beautiful. Now from PC, the electrons are sent to now. From PC, the electrons are sent to the next reaction center. We have one reaction center here. We also have the axillary pigments. Where did it vanish? Why is it vanishing? We have different reaction centers here. This is called as, again, this is also chlorophyll A. Chlorophyll A. Now this works at 700 nanometer. 700 nanometer. This is called as your PS1. This is called as PS1. Now from here also electrons are being transferred. Yes. From here again the electrons are sent up. Now from here electron is again taken up by a primary electron acceptor. The primary electron acceptor here is going to be FES, iron sulfur center. From iron sulfur center it's sent to FD, ferredoxin. That is ferredoxin. From ferredoxin finally the electrons are sent to your plus NADP plus. NADP plus is taking the electrons. It is taking the electrons. Two electrons. It is taking the electrons and also it is also taking 2H plus from the stroma. 2H plus from the stroma. So tell me, what do we call addition of protons? What do we call addition of protons, or addition of protons is called as reduction. So this NADPH is getting reduced to NADPH2. NADPH2. That is, and the enzyme enzyme which is involved here is FNR enzyme. Ferredoxin NADPH reductase enzyme. FNR enzyme is here. FNR enzyme is right here. Now in this entire cycle, is there ATP being produced? Yes. Between PQ to CY, here 1 ADP is getting converted to ATP. ATP is being produced. Yes. Now, if electrons are being consumed here continuously, do we require more electrons? Obviously, we need our electron reservoir, right? So, on the chlorophyll A, this side, in the lumen side, what is happening? Your H2O is splitting up. There is something called as photolysis of water is happening. That is H plus O2 plus electrons. Right? Plus electrons. Now what happens to this oxygen? This oxygen is released as a byproduct. It is removed outside. Byproduct is removed outside. Now what about two electrons? Electrons are sent here. Now what about this H plus? Now this H plus, main work of this H plus will be where? ATP synthesis. In ATP synthesis, this H plus is required. That is your entire non-cyclic photophosphorylation. I hope all of you wrote down. This. This is very important to understand the entire thing. Let me quickly summarize for you. We have chlorophyll A. Chlorophyll A works at 680 nanometer, which is PS number 2. That is your photosystem number 2. Sunlight is coming, falling on it, electrons are being excited. From here, electrons are taken up by primary electron acceptor, that is pheophytin. From pheophytin to plastoquinone to cytochrome BCF complex, then plastocyanin. From plastocyanin, electrons sent to chlorophyll A, that is P700, right? Photosystem 700. From here, electrons are again excited to taken up by the primary electron acceptor. Now, from here, from LFD, electrons are sent to your NADP plus. So if someone asks you, which is the final electron acceptor in your non-cyclic, the answer should be NAD plus. NADP plus is the final electron acceptor. It is the final electron acceptor. Awesome, right? Do all of you understand? This entire thing? It's such a beautiful thing, right? It's such a beautiful entire structure. Entire beautiful thing. This is called as Z scheme also. So can I write few points here for all of you to understand? First point here is, do we have oxygen evolution here? Yes. O2 is evolved here. I can say O2 is evolved here. Okay, O2 is evolved. Do we have photolysis of water here? Yes. We have photolysis of water. That is, water is being break down in the lumen side. I'll tell you where. What photolysis of water? ATP is being produced. Yes, sir. ATP is being produced. What about NADPH? Yes, sir. NADPH2 is also produced. Yes, NADPH is also produced. Now, whatever the electron is going here, is the electron coming back here? No, electron is not coming back. It's called as non-cyclic. And does it look like a Z shape? Here I have not done properly. It looks like a Z shape, right? Z shape. So it's also called as Z scheme. Called as what? Z scheme. Also called as what? Z scheme. Clear? Do all of you understand? Very beautiful it is. You can understand, you can solve so many questions, students. And if you trust me, this one particular slide will fetch you so many marks. If you truly understood every single step, and if you've written with me and you know it, so many marks are yours to grab here. So many marks. In fact, NEET 2024 question came from here. NEET 2024 question came from here only. Okay? Understood? This is your non-cyclic. Now what about cyclic? Is cyclic difficult or easy? Obviously. Cyclic is going to be even more easy. Cyclic is going to be even more easy. Because in cyclic we only have one photosystem. We only have one photosystem, that is PS1. PS1 works at 700 nanometer. 700 nanometer. Here also we have axillary pigments. Yes sir. Here also we have the certain accessory pigments. Accessory pigments are here. Then, so whenever we have the sunlight coming, all the accessory pigments will trap energy, and all the energy is given to the reaction center. All the energy is given to the reaction center. Now in the reaction center what happens? In the reaction center the electrons get excited. Electrons get excited. Two electrons are getting excited. Again electrons are taken up by primary electron acceptor, which is nothing but your FD here. FES. FES. Iron sulfur centers. It is iron sulfur centers. FES. From FES it goes to FD. From FD it goes to again PQ now. PQ to CY, CY to PC. What is it? Ferredoxin, plastoquinone, cytochrome BCF complex, and plastocyanin. From plastocyanin the electrons are again sent back to the photosystem 1. Now here, is NADPH produced here? No, NADPH is not produced. But is ATP produced here? Yes, ATP is produced here. ATP is produced here. ADP is getting converted to ATP. Between PQ and CY again, ADP is getting converted to ATP. Between PQ and CY and between FES. So this is called as your cyclic. It's called as cyclic. Now in cyclic, do you see any oxygen is evolved? No. NADH is produced? No. Photolysis of water is happening? No. Final electron acceptor? The final electron acceptor is going to be chlorophyll A itself. Chlorophyll A itself. Okay. That is your cyclic photophosphorylation. That is your cyclic photophosphorylation. So easy, right? So easy, right? Easy or difficult? Tell me in the chat. Easy or difficult? Only ATP is produced now here. Only ATP is produced. Only ATP is produced here. Only ATP. Clear? That is going to be your cyclic photophosphorylation. The next thing you have to learn here is a question. See this question now. This is a NEET 2024 question. I'll give you 1 minute. Take 1 minute and solve this question. See this. It says that the photochemical phase, that is also called as a light reaction phase of photosynthesis, includes, what did we have? What did we use? Tell me. Oxygen was released? Yes. Formation of ADP was there? Yes. Use of ATP? No. ATP is not used. ATP is produced. Splitting of water? Yes. Carboxylation? No. So A, B, D. Option number 2. Option number 2. That's all. In a light reaction, oxygen is released, NADPH is produced, ADP is not used, water splitting is there, carboxylation is not happening. So ABD answer. Easy question, very easy question. It cannot be 4. See ATP is not used anywhere. Is ATP used anywhere? No. ATP is produced. ATP is produced. Here also ATP is produced here. ATP is produced and NADP is also produced. It is only production, there is no utilization here. There is only production, there is no utilization here. Understood? This right here is a NEET 2024 question. Okay, let's quickly see the difference between cyclic and non-cyclic. In cyclic, electrons moves in a cycle. Yes, electron kind of moves in a cycle here. It linear path. Okay, only photosystem 1 is involved. Yes, we just now saw only photosystem 1 is involved. Here both photosystem 1 and 2 both are involved here. Electrons are first expelled from the reaction center 1. Yes, reaction center 1 is coming out. But in the case of your non-cyclic, From reaction center 2, obviously see here. From reaction center 2 it is coming out. Reaction center 2 or photosystem 2, right? Photosystem 2. Electrons return to the P700 after passing through the electron transport system. Yes, coming back to P700. But is it coming back here? No. It is electrons do not return back to P700. They are not coming back. Once gone, they are gone. Okay. Occurs in isolated chloroplast and photosynthesis bacteria. Okay. Here occurs in higher plants, algae and cyanobacteria. Oxygen is produced in the case of your non-cyclic. In the case of cyclic, there is no oxygen. No oxygen. Occurs in anaxogenic photosynthesis complex, no oxygen required. Oxygen is evolved here. Final electron acceptor is P700. Final electron acceptor is going to be NAD+. I told you. NAD+ in the case of your NAD+ case in the case of your NAD+ in the case of your non-cyclic. In the case of cyclic, final electron acceptor is chlorophyll 1. That is photosystem 1. Photosystem 1. Photolysis do not occur here. In the case of cyclic, in the case of non-cyclic, photolysis of water happens. That is, water molecules is broken down. Water is broken down. Easy. Students, all these concepts are going to be easy if you understand. If you just write, if you by heart, nothing is going to happen. See here, which of the following is not a product of your light dependent reaction of photosynthesis? Not a product of your light dependent reaction. O2 is there, we just now learned. Yes, O2 is there. ATP is produced. Yes. NADP is produced. Yes. Is glucose produced? Did you see glucose production light reaction anywhere? No, sir. Glucose is not produced. So what is the answer? Glucose. Glucose is the answer. That's all. Which of the following is correct regarding the cyclic photophosphorylation? Absolutely correct, they're telling you. It involves both PS1 and PS2. In cyclic, it involves both. Tell me, do you have both in the case of cyclic? PS1 and PS2 both are there? No. In the case of cyclic photophosphorylation is correct. So this is incorrect, right? It is incorrect. Correct. Electron pass through photosystem 1 are again cycled back to PS1? No. Correct. It is also incorrect. For electrons are from a second back. Yes, this is correct. They are going back and they are coming back. In a cycle, they are coming back. It requires external electron donor? No. It doesn't require electron donor because electrons are being cycled. Doesn't require external donor. Both ATP and NADP are produced? No. Both are not produced. Only ATP is produced. So which is correct? Only this is correct. Only this is correct. Okay. Clear all of you? Why B is the answer? See, in the case of your cyclic, we do not have ATP and NADPH. Both we do not have. It does not have both PS1 and PS2 does not have. It is not correct. Not correct. Not correct. Not correct. Okay. Now, can we start with the next concept? Can we start the next concept, friends? You are almost 50% of the chapter is actually over. Done. It's done, not done. Okay. We left with few more topics, that is chemiosmotic hypothesis, C3, C4, C2. Chapter is done. What is so difficult in this chapter? What is so difficult in this chapter, students? The next concept is going to be your chemiosmotic hypothesis. And before I tell you that, did I tell you here, everywhere did I tell you, ATP is produced here? Okay, ATP is produced. ATP is produced here? Yes, sir, ATP is produced. ATP is produced here also? Yes, ATP is produced. But how is ATP produced? How is the ATP even coming to the picture? From how is it coming? The answer to that question, how the ATP is produced, how can it be produced? Answer to that question is given by your chemiosmotic hypothesis. So chemiosmotic hypothesis tells you one thing. Chemiosmotic hypothesis tells you that how the ATP is produced. It tells you that how ATP is produced. See, synthesis of ATP is called as phosphorylation. Synthesis of ATP using light is called as photophosphorylation. I told you already. ATP synthesized in cyclic and non-cyclic photophosphorylation. Yes, it is synthesized. Chemiosmotic hypothesis explains the mechanism of synthesis of ATP. That's all. That's all. That is all chemiosmotic hypothesis is. Chemiosmotic hypothesis is explaining you how the ATP is being produced in cyclic and non-cyclic. Okay. Now, if you want to detailedly understand, you need to understand this particular diagram. It looks very scary, but you already know this diagram. Yes. Now, In non-cyclic photophosphorylation, non-cyclic, what did we have? Was there photolysis of water? Tell me in the chat. Was there photolysis of water in a non-cyclic? Yes or no in the chat? Tell me. Yes or no in the chat? Tell me. In the case of your non-cyclic photophosphorylation, was there photolysis of water in non-cyclic? You're like, yes sir, photolysis of water is there. Oxygen is being evolved in non-cyclic. If you see here, hear this. See this diagram right here. Let me tell you what this diagram is. Actually, this diagram is. What this entire diagram is. This is your thylakoids, right? Thylakoids. Entire stack is called as grana. When you take each individual thylakoid, and we zoom in, this is what we obtain. Each individual thylakoid, each individual coin-like structure, this entire structure here. It is the entire structure here. Each thylakoid. So this is the structure of your thylakoid. Now, didn't we learn on the thylakoid membrane we have both PS2 and PS1? Yes sir, you have been drawing from that time. We have PS2 and we also have the PS1 here, and non-cyclic photophosphorylation is happening here. Yes. See, what is this here? Did we learn this? This is photosystem 2, P680. Light is coming. Photolysis of water. It is sent into PQ. Plastoquinone. See, plastoquinone. We learned that. Then we have what? PQ, CY and PC. Didn't we learn that? PQ, CY, PC. We learned that, right? Plastoquinone, cytochrome B6F complex, capital B, okay. F. B6F complex and plastocyanin. This technically right now which is happening on the top is what? What is this? Non-cyclic photophosphorylation. Non-cyclic photophosphorylation is happening on top. Non-cyclic photophosphorylation. Okay. See here. Final product is going to be your NADPH. NADPH2. With the help of FNR, ferredoxin reductant complex. We learned that, right? Ferredoxin, NADPH reductase. Okay. Now, what is actually happening in your chemiosmotic hypothesis? If you want to understand that, for the entire process to take place, there are four main requirements. The first main requirement is called as your proton gradient. Second step is called as proton pump. Third thing is called as intact membrane. We need an intact membrane. Then we also need something called as one complex. It is also called as what? ATP synthase. It's also called as your ATP synthase is required. Four things are required for your entire chemiosmotic hypothesis to take place. Now, do we have proton pump here? Yes, we have proton pump here. Do we have intact membrane here? Yes, we have the intact membrane here. Do we have F0, FO1 complex? Yes, we have right here. We have right here the F0 and F1 complex. F1 complex. So the only thing we require right now, is the proton pump. We require the H plus gradient. Now, how is the H plus gradient produced here? There are three different ways. By which H plus gradient can be maintained or can be formed. Let me tell you one by one. The first one right here. When we were learning your non-cyclic photophosphorylation, didn't I tell you when water breaks down, it is producing O2, byproduct, plus electrons. Electrons are sent here, plus H plus. Now this H plus is sent here in the lumen side. So can I tell inside the lumen of thylakoid, there is increase in H plus? Yes, there is increase in H plus. Okay, increase in H plus. Now, can you see this plastoquinone? This plastoquinone is actually a hydrogen donor. It is a hydrogen donor. What will it do? Whenever the electrons are passing, it will use the energy of the electrons and it will transfer one proton from the stroma region and it will send the H plus into the lumen. So can I tell again, inside the lumen, H plus concentration is increasing. First one with the help of photooxidation of water, second one with the help of plastoquinone. Inside the lumen, H plus concentration is increasing. Can you see? Lumen high H plus. Okay, high H plus in the lumen. From here, from the stroma, within the lumen, it is coming, H plus is coming. So can I tell if H plus is coming from the stroma into the lumen, H plus is reducing in the stroma? Yes, sir, H plus is reducing in the stroma. Now, in the last step here, didn't we learn? When there is reduction, one H plus is used from the stroma. Didn't we learn that step right here? Somewhere we learned, right? In non-cyclic, if you go to the non-cyclic photophosphorylation right here, non-cyclic, didn't we learn here? 2 H plus from the stroma. That is taken up by electron and then it is getting reduced. Right? So, if you go back to this slide right here, since H plus is used from the stroma again here, in the last step of preparation of NADPH2, can I tell again H plus is being reduced in the stroma? H plus is again reduced in the stroma, right? So technically speaking, inside the lumen, H plus concentration is increasing. In the stroma region, H plus concentration is decreasing. Did we get a proton gradient? 3 different methods? Yes, sir. We got the proton gradient. Completely done. Proton gradient done. Now, you need to understand here is, once all the proton is here, H plus is here, in total of 4 H plus is required. Once 4 H plus goes, 3 H plus required for rotating the ring, 1 H plus to bring the phosphate. So, once 4 H plus pass through the F0 F1 complex, 1 ATP will be produced. That's all. That is called as chemiosmotic hypothesis. It tells you that how ATP is being produced. In your cyclic and non-cyclic photophosphorylation. How ATP is produced in your cyclic and non-cyclic photophosphorylation. Done, all of you? Done? This is called as chemiosmotic hypothesis. Entire points are given you. You can go through this once and it is going to be very easy to understand. The next concept is going to be dark reaction. We are coming towards the end of the chapter now. Light reaction is over. Light reaction is done. Now we are going to the dark reaction, the dark side of it. Now what happens in a dark reaction, students? So if all of you understood light reaction, if you do not have any doubts regarding light reaction, quickly greenhouse in the chat, which will confirm that yes, you know what is happening in your light reaction. Okay. Now let's go to the dark reactions now. Now, tell me, where does the dark reaction takes place? All of you are aware that the dark reaction takes place in the stroma region. Yes, it takes place in the stroma region. All of you know that, right? So, it's also called as, dark reaction is also called as your Calvin cycle. It is also called as light independent reaction because light is not required here. And it is the dark reaction is also called as your PCR cycle. Photosynthetic carbon reductase cycle. See here. Photosynthetic carbon reduction cycle. Not reductase, carbon reduction. Reductase is the enzyme. Photosynthetic carbon reduction cycle. PCR cycle. Photosynthesis. That is your dark reaction. Now, what is the product of your dark reaction? The product of a dark reaction is carbohydrates are the products. are the products of a dark reaction. Dark reaction. Okay? Of a dark reaction. Now, what is the raw material? What is the raw material? Obviously, the first raw material is ATP. Then we have NADPH. Both of these are coming from your light reaction. Both of these are coming from your light reaction. Light reaction. What about CO2? CO2 is coming from the air. How is it coming? Through stomata. Through stomata, it is entering. Through stomata, can you see this stomata? Through stomata, CO2 is entering inside. CO2 is entering inside. And we also know in the starting of the class, I told you, CO2 gets reduced to glucose. So CO2 is very important. CO2 is very important. And if you look at your C3 cycle, it's also called as, Calvin cycle is also called as your C3 cycle. It has three main steps. The first step is called as your carboxylation. We have carboxylation. Then we have reduction. And then we have regeneration. Three different steps here. Carboxylation, reduction, as well as regeneration. Now we will completely draw it and understand the entire three steps of it now. Entire three steps. So can we draw, all of you students? I want everyone to open your book. And write with me. Then only you will truly understand the dark reaction or the C3 cycle. Okay, ready all of you? C3 cycle or the dark reaction. Okay. So the C3 cycle or dark reaction starts with. The C3 cycle or the dark reaction starts with RUBP. RUBP. What is this? Rubulose 1,5 bisphosphate. 1,5 bisphosphate. Okay. Rubulose 1,5 bisphosphate. This RUBP, which is 5 carbon. It is 5 carbon compound. RUBP is a 5 carbon compound. It takes 1 carbon from the atmosphere. So CO2 is entering from the atmosphere. CO2 is entering. CO2 is how many carbon? 1 carbon. 1 carbon. Okay, 1 carbon. So 5 carbon plus 1 carbon, how much should be? 5 plus 1 is how much? Obviously, sir, you will tell me. So 5 plus 1 is 6. So we should have a 6 carbon compound. This is what all the scientists in the world were thinking. 5 carbon compound RUBP plus 1 carbon, you will get a 6 carbon compound. But that is not the case. That is not the case. You end up getting something called as PGA, phosphoglyceric acid. Phosphoglyceric acid, also called as your PGA. We get, in fact, this PGA right here is a 3 carbon compound. 3 carbon compound. But you get only one? No. You get 2 of them. So 3 2s are 6. So 6 carbon and 6 carbon, balance it out. And the first stable product, this right here, is the first stable product. Since the first stable product is 3 carbon, that is why it's called as what? That's why the cycle is called as C3 cycle. That's why we call it C3 cycle. The first step which we just discussed, is called as carboxylation. Carboxylation. Now in this carboxylation, is there any enzyme involved? Do we have certain enzymes working here? Yes, we have enzyme. We have the Rubisco enzyme. Here. Rubisco. Rubulose bisphosphate carboxylase oxygenase. Rubisco enzyme. This is called as carboxylation. Phosphoglyceric acid is produced. Now from here, your PGA gets converted to triose phosphate. Triose phosphate. Triose phosphate is also a 3 carbon compound. Yes. Now in the second step, students, is there utilization of ATP and NADPH? Yes. In the second step, there is utilization of 2 ATP here. 2 ATP gets converted to 2 ADP. The same reaction, 2 NADPH2 gets converted to 2 NAD plus. So NADPH2 and NAD plus are getting consumed here, 2 of them, and 2 ADP is getting consumed here. Now this second step is called as reduction. Called as your reduction. This is called as reduction stage. Reduction is happening. Done. Triose phosphate. Now this triose phosphate eventually becomes what? This triose phosphate eventually gets converted to glucose. Glucose. Yes, we have the glucose. We have the glucose. Done. Sorted. Right. Sorted. Now, in the last step, again, your triose phosphate gets converted back to RUPP. In this step also, one ATP is being consumed. One ATP gets converted to ADP. In this last step. And the last step is called as regeneration. It's called as what? Regeneration. So first we have carboxylation, reduction and regeneration. Carboxylation, RUBP to PGA, 2 molecules of PGA. Rubisco is the enzyme. 1 CO2 coming from here. So 5 plus 1, 6 carbon. Second step is called as reduction to form triose phosphate. Triose phosphate gets converted to glucose. In the reduction step, there is 2 ATPs being consumed, 2 NADPH is being consumed here. Done. Now regeneration, in regeneration also 1 ATP and 1 ADP is consumed. In your regeneration, 1 ATP is converted to 1 ADP. Now you tell me in the chat, how many total ATP did we consume here? The total ATP consumed here. 2 plus 1. For 1 CO2, we have total of 3 ATP being consumed. 3 ATP as well as 2 molecules of NADPH. NADPH2, 2 molecules of NADPH2 and 3 molecules of ATP is being consumed. 3 ATP, 2 plus 1, 3 ATP and 2 NADPH. This is the entire C3 cycle. Students. C3 cycle. Now, in the examination they can ask you, they will not ask you for 1 CO2. Imagine they ask you for glucose. Then what should you do? If they ask you for glucose, tell me, glucose is how many carbon? 6 carbon. Yes, glucose is C6H12O6. So you just multiply into 6. 18. 6 is 18 ATP. 12 NADPH. Done. Just multiplied that time. Done. Done, right? Easy. 18 ATP and 12 NADPH for glucose. Such an easy concept, students. C3 cycle. Yet so many questions come from here. So 12 NADPH and 18 ATP and 12 NADPH. Now examples for your C3 cycle. Yes, we have oats, soybean and wheat. All of these are examples for your C3 cycle. Now, did we learn in carboxylation? There was an enzyme. In carboxylation, the enzyme was Rubisco enzyme, which is also the most abundant enzyme. Yes, most abundant enzyme in the world. Abundant protein in the world. Now, this Rubisco enzyme has carboxylase. So, this will form a bond with RUBP plus CO2, which will lead to a normal C3 cycle. Now, sometimes what happens is, instead of carboxylase, oxygenase will be activated. I'll tell you when it is activated also. Oxygen is activated and RUBP forms a bond with oxygen, and that will lead to C2 cycle. That C2 cycle is called as what? C2 cycle is called as photorespiration. So what happens in photorespiration? In your photorespiration, students, in your photorespiration, RUBP is combining with oxygen to produce a 2-carbon compound and 3-carbon compound, which we'll tell you in next minute, with the help of Rubisco enzyme because of oxygenase activity. Because of oxygenase activity. That is called as your C2 cycle, also called as your photorespiration. Also called as photorespiration. See, it occurs in C3 plants. O2 is consumed and CO2 is released in the process. O2 is consumed. Rubisco oxygenates RUBP. It oxygenates RUBP. Okay, oxygenates RUBP. Now, what exactly happens, students? What exactly happens here? In your photorespiration, RUBP forms a bond with oxygen. Now, you tell me, this is 5 carbon, no carbon here, 0 carbon. Like carbon 0, no carbon here, no carbon here. So this will release in the this will give a result into the product. We get PGA. Along with PGA, we also get something called as PG. PG. Now what is this PG? PG is nothing but phosphoglycolate. Phosphoglycolate. This PG is a 2-carbon compound. This PGA is 3-carbon compound. 3 plus 2 is 5-carbon compound. See, carbon balance is maintained. Carbon balance is completely maintained. Now, eventually what happens? Eventually, this PG gets converted to PGA. Eventually, it gets converted to PGA. But in this step, in this entire step, did you see any ATP being released out? No. So in this entire step, forget ATP. In this entire step, did you see any sugar formation? No sugar. No sugar formation. Did you see any sugar formation here? No sugar formation. Did you see any ATP formation? No. Did you see but ATP is being consumed here? Yes, ATP is being consumed here. ATP is being consumed in this reaction. In phosphoglycolate, ATP is being consumed. In this step, since oxygen is binding to here, CO2 is released outside. CO2 is released outside, which is very bad for the plant. So when so many things are happening, and none of them is beneficial, none of them are beneficial to the plant, and that's why this is called as what? Waste. It is called as a wasteful process. No sugar, ATP is being consumed, CO2 is being released out. Waste of time, waste of money, everything. And it only happens in your C3 plants. It only happens in your C3 plants. Only in your C3 plants. Only in your C3 plants. Done? Now, when does it happen? When will the C2 cycle occur? This is very important. Will it occur all the time? Will the C2 cycle occur all the time? No. C2 cycle will occur at only few places where oxygen is very high. When the oxygen is very high, Rubisco enzyme will favor oxygen and not bind with CO2. So when oxygen is very high, CO2 cycle will happen. Or when CO2 is very low. Obviously when CO2 is very low. Alright. It also happens in high light conditions. High light and temperature condition. When there is high light and high temperature, this happens. High light and high temperatures, low CO2 and high oxygen. In these three following times, your C2 cycle will occur. C2 cycle will take place. Clear all of you? Clear all of you? At these three places, when there is more oxygen, when there is less CO2, and when there is high temperature and high light, C2 cycle will get activated. That's called as photorespiration. Will get activated. But does this happen in your C4 plants? Does this wasteful process C2 cycle, does it happen in your C4 plants? Absolutely not. That is, this photorespiration does not take place in C4 plants. Not at all. It doesn't happen. It doesn't happen in your C4 plants because C4 plants are very smart. Your C4 plants are very, very smart. They're played very smartly because they have a special structure. They have a certain very special structure called as your Kranz anatomy. they have a special structure called as your Kranz anatomy. This is your Kranz anatomy. This is your Kranz anatomy. If you look at your Kranz anatomy, all the vascular bundles, can you see this? All the vascular bundles are arranged in a wreath-like structure. All the vascular bundles are arranged in a wreath-like structure with large chloroplasts. In a wreath-like structure, in a large chloroplast with no intercellular space here. Absolutely no. There is no intercellular space. No intercellular space. Nothing. If there is no intercellular space, can anything enter inside? Can any type of gas enter inside? No. Absolutely. No gas can enter inside. No gas can enter inside. They are very smart plants. Very smart. No gas can enter inside. Now NCI tells you wreath-like structure. Do you know what is a wreath, students? Do any one of you know what is wreath? Wreath. Have you, any one of you heard this? What is the meaning of wreath? All of you have seen this. You might not remember, that's all. This right here is wreath. Have you seen this Christmas? In Christmas time they add, right? Christmas time they add this. This is called as wreath. In English, this is called as a wreath. Does it look like that? Does it look like a wreath-like structure now? Yes, it looks like a wreath-like structure. Wreath-like a structure. Done. This is wreath. See here. Leaf anatomy of C4 plant is called as Kranz anatomy. Bundle sheath cells are arranged in the form of wreath around the vascular bundles. Bundle sheath cells are characterized by the having a large number of chloroplasts, thick walls, and no intercellular space. Absolutely absent. Absolutely absent. They can tolerate high temperatures, yes. Respond to high light intensity, yes. Lack photorespiration completely. No photorespiration at all in your C4 plants. And also greater productivity of biomass. Why greater productivity? If there is no photorespiration happening, only C3 cycle is taking place inside. Only photosynthesis is happening. So more productivity. That's all. More productivity here. And this C4 cycle is also called as Hatch and Sack pathway. Hatch and Slack pathway, because these were the two scientists who discovered this pathway. Hatch and Slack pathway. Okay, Hatch and Slack pathway. Done. Now, I want everyone to open your books and quickly write down the entire C4 cycle. Entire C4 cycle, also called as Hatch and Slack pathway. Hatch and Slack pathway. Okay. So in your C4 cycle, we have two type of cells. In your C4 cycle, we have two type of cells. One is called as your bundle sheath cells. Bundle sheath cells. Okay. Other one is called as mesophyll cells. Mesophyll cells. Now, what exactly happens in your bundle sheath cell and mesophyll cell? Let's understand. In your bundle sheath cells, what happens is CO2 is entering inside. Okay. Because bundle sheet is a mesophyll present. See here. Mesophyll cells are present on the outside. Can you see? Mesophyll cells are present on the outside. Mesophyll cells are present on the outside. So your gas can enter inside. Yes, gas can enter inside. Yes. CO2 is entering. Now here, in this case, CO2 plus phosphoenol pyruvate. In the case of your C3 cycle, it was Rubisco. But here we have phosphoenol pyruvate, also called as PEP. Your phosphoenol pyruvate plus your CO2, 3 carbon, 1 carbon, will result in OAA, oxaloacetic acid. Oxaloacetic acid, which is a 4 carbon compound. Since the first stable product is a 4 carbon compound, it is called as C4 cycle. It is called as C4 cycle. Now this oxaloacetic acid gets converted to malic acid. Malic acid, which is also 4 carbon. Now this malic acid is sent to your bundle sheet. Malic acid. Malic acid. Now this malic acid gets converted to what? This malic acid gets converted to your oxaloacetic acid again. 4 carbon, 4 carbon. This oxaloacetic acid gets converted to pyruvate. Pyruvate, which is around 3 carbon. 3 carbon. 4 carbon, 3 carbon. 1 carbon is missing. That's right. 1 carbon is released outside. 1 carbon is released outside. Now this carbon right here, students, this carbon right here will enter, it will enter the C3 cycle inside. It will enter the C3 cycle. It will enter the C3 cycle. Can you see? CO2 is entering the C3 cycle. Now again, what happens in C3 cycle, students? Inside your C3 cycle, Rubisco is there. Rubisco is there. We have RUBP. RUBP forms bond with Rubisco, and we get your same product again, PGA. What happens here in C3 cycle? We have RUBP plus CO2 from here. We will get your PGA. Same thing will happen inside. Now, but entirely this oxaloacetic acid is converted to pyruvate. This pyruvate will be sent again back to on top. This pyruvate will again be sent back to here, and this pyruvate once again sent back here, and this pyruvate will again get converted to PEP. We have pyruvate here, 3 carbon. Then this pyruvate gets converted back to PE. This pyruvate again gets converted back to PEP. That is phosphoenol pyruvate. And in this step right here, 2 ATP is being consumed. 2 ATP gets converted to 2 ADP. 2 ADP to 2 ADP. Clear? Senorita, just follow this. You will be able to solve all the questions. So what do we have? First we have, in the starting we had CO2 entering, CO2 plus phosphoenol pyruvate, 3 carbon plus 1 carbon, 4 carbon, oxaloacetic acid. Oxaloacetic acid gets converted to malic acid. Malic acid is sent to your bundle sheet. Now from bundle sheet, malic acid gets converted to oxaloacetic acid. Oxaloacetic acid again gets converted to pyruvate. 4 carbon, 3 carbon, 1 carbon is missing. Then 1 carbon is released in the form of CO2. CO2 is entering the C3 cycle. CO2 is entering the C3 cycle. Now this pyruvate again is sent back to your mesophyll cell. From mesophyll cell, pyruvate is again getting converted back to phosphoenol pyruvate. That is your entire C4 cycle. Now within the C4 cycle, we have the what? We have the C3 cycle also. Since oxaloacetic acid is first product, C4, that's why the C4 cycle. This is your entire C4 cycle, students. Very easy to understand. Very easy to understand. For example, this is a table which you have to master right now. For example, cell type in which Calvin cycle takes place. In the case of your C3 cycle, mesophyll cell. In the case of your C4 cycle, bundle sheet cell. See, we learned here, right? In bundle sheet cell, C3 cycle is happening. Done. Cell type in which initial carboxylation reaction takes place. Here mesophyll cell, here also mesophyll cell. Initial fixation is happening in mesophyll cell. There we only have one cell, that is mesophyll cell only in C3. Okay. Now, how many cell types are there? 1, here we have 2. Which is the primary CO2 acceptor? Here we have RUBP. Primary CO2 acceptor in case of C3 cycle. Here we have PEP, that is phosphoenol pyruvate. The one thing which I didn't mention here is the enzyme. Here we have the Rubisco enzyme in C3 cycle. Here we have an enzyme called as pepcase. Pepcase is in there. Pepcase is the enzyme there. Pepcase. Okay, pepcase is the enzyme. See, number of carbons in the primary electron acceptor, primary CO2 acceptor we have. RUBP is 5. Your phosphoenol pyruvate is 3 carbon. It will be right here. 3 carbon. In C3 cycle it was 5 carbon. RUBP. Then what do we have? Which is the primary CO2 fixation product? PGA is the first product here, which is 3 carbon. That's why the C3 cycle. Here we have oxaloacetic acid, which is a 4 carbon compound. Hence it's called a C cycle. Okay. Now, does the plant have Rubisco? Yes, both have the Rubisco. Does the plant have pepcase? No. In your C3 cycle, there is no pepcase because there is no requirement, right? Here we have pepcase. Yes, pepcase is there. Which cell in the plant have Rubisco? Here we have mesophyll cell. But in the case of your C4 plants, it is bundle sheet cell. See here. Bundle sheet cell will have your Rubisco enzyme. Right? CO2 fixation under high light condition, it's medium. Here it will work perfectly. High light condition will work perfectly. Whether photorespiration is present at low light, obviously, low light there will be no photorespiration because it is not required only, no photorespiration. But under highlight condition, here we have photorespiration. We have photorespiration in your, photorespiration is present in the case of C3 cycle. But in the case of C4 cycle, do we have photorespiration? No. We never have photorespiration in the case of C4 cycle, never ever, never. Okay. Now, whether photorespiration would be present at low CO2 concentration? No. Here we will not have any, but at low concentration we have photorespiration here. I told you, right? When does the C2 cycle occur? When there is high O2, when there is high O2 or very low CO2, right? Very low CO2 present. Whether photorespiration would be present at high CO2? No. At high CO2, why photorespiration? Plant can take the CO2 and work. No photorespiration in both. No photorespiration in both. See here, temperature optimum 20-25 degrees Celsius. We have 30-40 degrees Celsius here. Examples: wheat and rice. Example is maize, sugarcane and sorghum. Maize, sugarcane and sorghum. That is the end of your photorespiration. C3 cycle, C4 cycle. Done. What is the answer? See this. Light independent reaction of photosystem occurs in. Light independent reaction is called as what? Dark reaction. Dark reaction happens in stroma. Dark reaction happens in stroma. Carboxylation in C3 plants require the following enzyme. Carboxylation requires which enzyme? Obviously, carboxylation in C3 cycle requires Rubisco enzyme, right? It requires Rubisco enzyme, C3 cycle. Look at this question now from your NEET 2024. Which of the following are required for dark reaction photosynthesis? Do they require light? No, not required. Chlorophyll required? Not required. CO2 required? Yes, required. ATP required? Required. NADPH required? Required. Clear? What is the answer? PE. CDE, right? CDE. CDE is the answer. CDE. Done. The products of light reaction. These are the products of light reaction. Products of light reaction, which are required in your dark reaction. CO2 coming from the air. Now, see this question. Again, 2024 question. Again, 2024 question. How many molecules of ATP and NADPH are required for every molecule of CO2 fixed in your Kelvin cycle? Fixed in your Kelvin cycle. Let's see the answer. See here. Every Kelvin cycle we require 3 ATP and 2 NADPH. Very easy. Very, very easy. I don't know. My student, if you were my student last year, you would have answered this question very easily. Where do you have 3 ATP? 3 ATP and 2 molecules of NADPH. Answer is here. You don't need to think twice now. You know the answer, you know the concept. You written down the notes. Everything is in your hands. Everything is in your hands. Done. The last concept, which is around 2 pages in your NCERT, the last concept for today. The last concept for today is going to be, hello Sebastian. The last concept for today is going to be something called as your factors affecting photosynthesis. First of all, there is something called as internal factors. Something called as internal factors, which could be your number of age of leaf, orientation of leaf, shape of number of mesophyll cells, more photosynthesis, number of chloroplasts, more photosynthesis, amount of chlorophyll, more photosynthesis. These are called as your internal factors. These are called as internal factors. All of these factors depend on genetics. But we also have certain external factors. Before we understand the external factors, there is something called as Blackman's law of limiting factor. What is this limiting factor, students? What is this limiting factor? Let me explain to you in a beautiful manner. You will never forget. You will never forget this. Okay. Limiting factor tells you that when many factors are involved in a biochemical reaction, such as photosynthesis, then then speed of reaction, speed or rate of reaction mainly depends on depends on that factor which is present in least amount. That's all. Many factors are involved in biochemical reaction, such as photosynthesis. Then the speed or the rate of reaction mainly depends on that factor which is present in the least amount. On that particular factor, the entire photosynthesis will depend. For example, quick example. In the winter season, early morning, light is very less. Yes, light is very less. So light becomes a limiting reagent. Light becomes what? Light will become a limiting factor. It is present in least amount. It becomes limiting factor. Yes, limiting factor. But what about in the case of summer? Summer again. Even in summer, early morning. Light is obviously a limiting factor. Early morning, light is a limiting factor. But as the day is progressing, do we have enough of light in summer? Yes, light is there in summer. But do we also know that in summer, water is very limited, moisture in the air is not there. So there is very little water. Yes, very little water. In that case, from light, water becomes the limiting factor. Water will become the limiting factor because water is present in the least amount now. So water becomes the limiting factor then. That's it. So limiting factor is the factor, or law of limiting factor is whichever the factor is present in the least amount, that becomes a limiting factor. That's it. Clear? Now we have external factors such as light, carbon dioxide content, temperature and water. Let's understand one by one. For example, in light, we have many categories, different parts. There are three main categories. The first category is called as your duration of light. All of you know we have short day plants, long day plants. Each of them have their specific requirements, right? Long day plants require more sunlight, short day require less sunlight. Similarly, when you have light quality, we have learned throughout the chapter we have learned that in blue regions and red regions, that is the high quality light at which photosynthesis is much more higher. Yes, we have learned that. But there is most important point. The most important one is going to be something called as light intensity. It is called as light intensity. The most important one. Because the light intensity controls the entire photosynthesis. It controls the entire photosynthesis. Light intensity. For example, if you look at this graph, right here, from O to A, this is O. From O to A, can I tell, as the light intensity is increasing, the rate of photosynthesis is also increasing? So can I tell light intensity is directly proportionate to rate of photosynthesis? It is directly proportionate. Yes. But after a certain point, can you see, from B to C, it becomes constant. It means that what? At only up to certain limit, it is a limiting factor. After some point, it is not a limiting factor. That is, saturation is required. Saturation rate. Now, what is the meaning of saturation here? The meaning of saturation is very simple here. Plants require only 10% of total sunlight. 10% total sunlight. That's all is enough for the plant. So in many cases, students, in many cases, light is actually very rarely, it is very rarely the limiting factor. It is very rarely the limiting factor. Very rarely. It is very rarely limiting factor. It is not usually limiting factor. It might be limiting factor only in some cases, such as your dense forest. You know, in certain dense forest, yes. In deep forest, it might be a limiting factor. But generally speaking, light is not a limiting factor at all. In fact, if light becomes too much, it is very dangerous. It is very dangerous to the plant. Okay. Now that reminds me, I think we missed out one point here. When we were talking about your photosystem, when we were talking about your light harvesting complex, what is the function of light harvesting complex? What is the function? What is the main function of light harvesting complex? I forgot to mention that point, and now I remembered. Light harvesting complex, right? Light harvesting complex. Your light harvesting complex has two main functions. Can you see? Because of your axillary pigments, the range of absorption is increased. So can I tell? See, it's not mentioned here, is it? I thought it is mentioned there somewhere. See, it's mentioned here. Absorption light and transfer to the chlorophyll A. The range is increased. The range is completely increased. The range is widened. The range of absorption of light is widened completely. Range is widened. Clear? Range is widened. And also, your axillary pigments protect the chlorophyll A from photooxidation. Imagine if all the light goes to your chlorophyll A, will it get damaged? Yes, it will get damaged. So your entire different small your axillary pigments, they'll absorb the light and then they'll give it to it. So it will protecting the chlorophyll A from photooxidation. 2 points we missed out. It's fine. Somewhere down the line I could remember, because we were learning about your limiting factors, right? Limiting factor. Clear? All of you? So limiting factor will directly proportionate only up to a certain limit, but after that it becomes constant. That is, it is no more a limiting reagent. Very rarely limiting factor, light. Okay, very rarely a limiting factor. Now what about your carbon dioxide? Carbon dioxide is a good limiting factor. It is a major limiting factor in the photosynthesis. Why? Because in the atmosphere, we just have around 0.03% to 0.04% of carbon dioxide in the atmosphere. And that is why it will become, obviously it will become a limiting factor then. Right? Limiting factor. And also students, if it is more than 0.05, if the CO2 concentration becomes more than 0.05, it can cause, it can cause damage. If it becomes more than 0.05, it can cause damage to the plant also. Now, at low light intensities, C3 and C4 plants respond to high CO2 conditions. Okay. In low light, they'll work fine. They'll work beautifully. Okay. In low light intensity, it doesn't work. Sorry. In low light, it doesn't work. In low light, they don't work. Neither. They do not respond at all. Why don't they respond? Because in low light, it is the limiting factor, right? It is the limiting factor here. In low light condition, if there is no light only, both of them will not work very well. Okay. In high light intensities, both C3 and C4 plants show increased rate of photosynthesis. In high light intensities, both will work properly. But very high, your C3 cannot work. Very high C3 cannot work. Okay. Very high C3 cannot work. But your C3 plants and C4 plants, they react very differently to CO2. In fact, your C3 plants, they have higher saturation. Your C3 plants show a saturation beyond 450 microliter per ppm. I can say ppm also. While your C4 plants has just saturation is around 360. Now, what is the meaning of saturation here? The meaning of saturation is very simple here. C3 plants can take up more CO2. Yes. Your C3 plants can take up more CO2. If the C3 plants can take more CO2, if more CO2 is there, can I tell? More photosynthesis. Yes, more photosynthesis. If there is more photosynthesis, can I tell? More productivity. More productivity. That is, more glucose produce, more productivity. It means that if you take a plant and you keep it under high CO2 concentration, you will get better results. Yes. And that is what we do. We call it greenhouse. We call it greenhouse. Greenhouse. Under greenhouse, in your greenhouse, what happens? CO2 is trapped inside. And inside the greenhouse, there is very high CO2. If there is very high CO2, more CO2, more photosynthesis, more productivity. Example is tomato and bell pepper. Example is tomato and bell pepper, which produce high productivity under high CO2 condition. Under high CO2 conditions. Understood? Next one is temperature. Temperature is very easy, students. Temperature is very, very easy. Most of your enzymatic reaction, it could be on dark reaction, Rubisco, Pepcase enzymes, they work at a very optimum temperature. Beyond that, they cannot work. Beyond that, they cannot work. Okay. And C3 plants, all of you should know, C4 plants respond to high temperatures and show increased rate of photosynthesis. So in the case of C4 plants, they will work very well. They will work very well. But what about C3 plants? C3 plants at high temperatures cannot work. C3 plants at low temperature or optimum than C4 plants. Tropical plants have, C3 plants cannot work. Okay. Tropical plants have high temperature optimum than temperate forest. That is very common point, obviously. Tropical will have higher temperature requirement. The last one is water. The last one is water. Your water actually plays a rather indirect role. I can tell it rather plays a very indirect role. Rather plays a very indirect role. What is the meaning, sir? I'll tell you. Let's take two cases. Take two cases. If there is less water, will the stomata close? Yes. To save transpiration, from the transpiration loss, stomata will close. Stomata is closing. Stomata is closing. Stomata close. If stomata is closing, CO2 entering inside also is becoming less. Yes. CO2 is not entering inside. If CO2 is not entering inside, photosynthesis is obviously decreased completely. Photosynthesis decreased completely. Simple logic. Logic. Right? Again, if there is less CO2, if there is less water, all the leaves will fall off. Can we see something called as wilting of leaf? All the leaf will try to curl. Yes, the leaf will try to curl. So if the leaf is curling or wilting, can I tell the surface area is also reducing? Is reduced. Surface area is also reduced. Yes, surface area is also reduced. If surface area is actually leaf only reduced, can I tell photosynthesis is reduced? Photosynthesis is reduced completely. Yes. Done. Done, students. That is your water. See this question here. See this question. This question. NEET 2024. In C3 plants, some oxygen binds with Rubisco and CO2 fixation is decreased. That is called as photorespiration. Okay. In C4 plants, mesophyll cells show very little photorespiration, while your bundle sheet cells do not show photorespiration. We know even in the case of mesophyll cell, there is no photorespiration. C4 plants, no photorespiration at all. So what is the answer? Both statement number 1 and statement number false? No. Statement number 1 is true, but 2 is false? Yes. 1 is true and 2 is false. 1 is true and 2 is false. Done. Entire notes will be given on the Telegram channel and also on my Telegram channel. That is going to be your Basavaraj Sir Biology. On this Telegram channel, all the notes will be available to you. All the notes will be available to you. Now students, I made you solve some questions. I made you solve some questions right now. Yes sir, we solved some questions. Now what if you want to solve more questions? What if you want to solve more questions? How do you do this? If you go to description link, if you go to description right here, there's one link right here which says year-long test series. Year-long test series. If you go to this page right now, you will have year-long test series coming entire year. For entire year, you will have test series. You will have total of 7 full test series, 6 subjects. As you have 12 part test series will cover your topics in detail. Physics, chemistry, biology, all topics covered. And in this test series, you will have entire thing. You realize 7 test series and 12 part test series. And the price for this right now is going to be around 2000 rupees. That is the price for this test series. And this is going to be there for entire year. So if you want to solve more questions, if you want to solve more mock tests and you want to be involved in the test series, that from now on you want to start with active revision, this is where you go. Okay, this is where you buy a test series. Okay, students. So with that being said, your entire chapter is over. And in the starting of the class, I told you, 2.5 hours back, I told you this chapter is the easiest chapter of all time. And students who have watched till the very end, you will tell me, yes sir, easiest chapter of all time. Every single concept I understood, every single question I'll be able to solve now. Every single question. So with that being said, I'll take off right now. All of you, 3 things always in my class. I'll always tell you 3 things to follow. After the class, revise whatever you have learned in the class. Read the NCERT. And the last point is solve questions. Follow these 3 things throughout the year. No one can stop you from clearing NEET. So with that being said, take care. Bye bye students.